This is the fourth and final video in Module 1, Introduction to Biochemistry. In this video, review of acid-base chemistry, I'm going to go over some of the properties of acid-base chemistry that are relevant to understanding biochemistry and how the pH of the surrounding environment influences biochemical molecules. So I know what some of you are thinking right now. You may be feeling worried about this subject. Traditionally, I have found that many students are worried about acid-base chemistry, but I'm here to say that it's actually really important for you to understand how acids and bases affect the pH of a surrounding environment, because that environment will affect the structure and activity of different biological molecules, particularly proteins. So it's really important to sort of get a baseline understanding of this. So I'm going to go through just a quick review of acid-base chemistry, and we're going to come back to this topic again when we talk about amino acids in the next module. But don't, I would go through some of your chemistry notes about this, your general chemistry notes about this, and ask as many questions as you can about this topic so to make you feel a little bit less nervous about acid-base chemistry. But again, this is really important for understanding biochemistry because many biological molecules have functional groups attached to them that undergo different types of acid or base reactions. For example, amino acids, each amino acids has a unique functional group. And so there are 20 amino acids, each with a unique functional group. Some of them are polar, some of them are nonpolar, some are charged, some are not charged. And so these types of molecules can undergo different states of protonation or different states of charge, depending on the environment they're in. It also will affect dramatically how those proteins fold, the interactions between these functional groups. And interactions between the functional groups will depend very heavily on their charge, and their charge depends heavily on the pH of the surrounding environment. In fact, proteins will denature or go from a native state to a denatured state, an inactive state, if they're placed in a pH that is outside their optimum pH environment. And so to give you an example of that, I've put a graph here that shows 3 different types of proteins. They all do somewhat similar jobs. They're all hydrolytic proteins, and they are involved in different stages of digestion. Pepsin is a digestive enzyme that breaks down proteins in the stomach. Salivary amylase is a digestive enzyme found in saliva that converts starches, complex starches, to simpler sugars. And arginase is a urea cycle enzyme that helps to convert or helps to produce urea during protein metabolism or catabolism specifically. And so you notice that each of these proteins, while they're all hydrolytic and and they're all involved in some type of metabolic pathway or metabolic pathway, they have really dramatically different optimum pHs. And so here, the optimum pH for pepsin is really low, and that makes a lot of sense. Pepsin is found in the stomach, where the pH of the environment is very low. And so if you place pepsin in the optimum pH for, say, salivary amylase, which is found in the saliva, pepsin will actually unfold because it's outside of its normal pH. And the reason why it unfolds is because the pH of the environment is affecting the charge of the functional groups that are included in the peptide structure of this protein. And so if you affect the charge of those functional groups, they can't interact appropriately in their folded state, and so they will unfold. And that is true for each of these proteins. If you place salivary amylase at a low pH, it too will unfold and become inactive. And so it's really important to understand the pH of the environment, since that's going to definitely affect the structure of enzymes, proteins, structural proteins, lipid bilayers, and virtually all biological molecules. Okay, so as a reminder, when I talk about an acid-base reaction, what I'm talking about is when an acid and a base are placed together, they react to neutralize the acid and base properties and produce salt and water as a response. So the cation of the acid, usually a hydrogen ion, cation of the acid will combine with the anion or the negatively charged OH group of a base to form water. The compound formed by the cation of the base and the anion of the acid is called a salt. And so one example of this would be hydrochloric acid, or HCl, which is a strong acid, plus NaOH, a strong base, will yield H2O and salt, or sodium chloride, NaCl. And so the acid and base properties of the HCl and the NaOH, respectively, have been neutralized to form a salt. So this is all really important. All of this is taking place in an aqueous environment, or a water-based environment. The pH of an aqueous solution, it depends on, or it's defined as the deionization of water. And so ionization, to refresh our memory, is an atom or molecule that acquires a negative or positive charge by gaining or losing electrons to form ions. And so when we talk about the ionization of water, water is split into its cation, hydrogen, and its anion, OH group base. I talked briefly about the equilibrium constant in a previous module, but basically we can redefine this equilibrium constant. So that would be the concentration of hydrogen ions in OH anions divided by the concentration of water. And this will define the position of this equilibrium reaction, of this deionization, of this ionization of water. We can replace this value with a different value called the ionization constant of water, which is specific to water itself. And that value, without getting into the derivation of these equations, is 10 to the minus 14. So the product of the hydrogen cations and the OH anions together in aqueous solution must always be equal to this value. And it's this right here that defines what's called the pH scale. In an aqueous solution, any changes in the cations are balanced by changes in the anions. And so together the product of these two concentration values will always equal 10 to the minus 14. And so what this graph is showing you is that down here we have pH on this axis, and then we have the ion concentration of either hydrogen ions or the OH anions on this y-axis here. And so if we, neutral pH would be where the concentration of hydrogen ions exactly equals the concentration of OH ions. And so that's a neutral solution right here at a pH of 7, or the product of H and OH equaling 10 to the minus 7. I'm sorry, no, not the product, my mistake. These are equal to each other, and so that equals 10 to the minus 7. I'm sorry about that. At a pH lower than 7, we consider that acidic. And so in this situation, there are more hydrogen ions than there are OH ions. And still the product of these two values will equal 10 to the minus 7. And so if there's an increase in hydrogen ions, as is the case with low pH or very acidic pH, there is a balanced decrease in OH ions. As pH increases to a more basic number, what this means is that OH ions are increasing, and so by default, H plus ions will decrease. And so pH is defined by the concentration of hydrogen ions. And because these two values are related to each other, you can talk about the opposite scale, or the inverted scale of the pH scale, which would be the POH scale. But we have to kind of pick one. And so we're talking more about the concentration of hydrogen ions when we talk about pH. And so pH is equal to the negative log of the hydrogen ion concentration in this aqueous solution. And so we're really only concerned about the hydrogen ion concentration, since if you know that, you can automatically determine what the OH ion concentration would be. And so here's our pH scale. It goes from 0 to 14. Anything below 7 would be considered acidic. And then as you get to a lower and lower number, it's increasingly acidic. Anything above 7 would be considered basic, and as you go higher and higher, it's considered increasingly basic. Strong acids are all the way down here at the bottom of the pH scale, representing the most acidic environment. And strong bases are up here at the top of the pH scale, representing the most basic environment. And then we have a couple of examples of different types of acids and bases and neutral solutions on this scale. Biological pH is considered to be somewhere slightly above 7, so like 7.2. And so that would be human blood, the bloodstream, human tears are fairly neutral in acidity. That's not true for every area of the human body. So for example, the stomach has a very low pH. And so that's represented here by gastric juice. That's stomach acid, will be a very low pH. But in terms of the human bloodstream, we consider that to be physiological pH, which would be more neutral. Milk and saliva also are a little bit more neutral, but they are below, a little bit more acidic than blood and tears. Okay. All right, and so acids and bases act together to alter the pH of a solution or to alter the hydrogen ion concentration of a solution. Acids are proton donors, and so they donate hydrogen protons. Bases are proton acceptors. They accept hydrogen ions to bind with the base molecule. Okay, and so these two can act together to alter the hydrogen ion concentration of a solution. If you add more acid, hydrogen ion concentration is going to increase because you're basically, when you're adding acid, you're adding protons. If you add more base, pH will increase, so the pH of a solution gets higher. And so when you add base, what's happening is any free hydrogen ions in that solution are binding with the base, as bases are proton acceptors. And so the hydrogen ion concentration in the solution will decrease and OH will increase. Strong acids and bases ionize completely in water. And so for example, what that means is if I were to add hydrochloric acid to water, the resulting reaction would be the production of hydrogen ions and chloride ions. This isn't true for weak acids and weak bases. They don't completely ionize in water. Their ionization rates depend on the pH of the surrounding environment that they are in. So what other else is happening in that water? So the HCl in this case is donating a proton, right? It's hydrogen is coming off, which increases the hydrogen ion concentration of the overall solution and will decrease or drop the pH. So just to kind of relate that, if you're putting hydrochloric acid in water, it deionizes and is contributing more hydrogen ions to the surrounding solution. And so more hydrogen ions means that we are more acidic and the pH is low. Weak acids and bases are more common in biological systems than strong acids and bases. And these really take the form of amino acids. So all of the amino acids that make up our proteins are acids, but they're weak acids. And they do not completely deionize in water, or they don't completely ionize in water. And so we have to sort of come up with a metric to measure the tendency or the probability that an acid is going to lose its proton when placed in an aqueous environment. And so the tendency of an acid to lose its proton is defined by the equilibrium constant. And because we're talking about acids specifically, we call these the acid dissociation constant, or the Ka. And so again, the equilibrium constant is the concentration of the products divided by the concentration of the reactants. So this is related to the video where I talked about thermodynamics. And so this is the equilibrium constant. We redefine that to be Ka because we're talking about an acid specifically. And remember that when we place an acid in water, it will dissociate Not completely, but it will dissociate into its acidic or its acid component, its hydrogen ion, and its conjugate base, which is what's left after the proton has been removed from the molecule. And so A could represent, any number of functional groups that are attached to this proton. If we want to measure, again, the tendency of that acid to lose its proton, we take the inverse log of the equilibrium constant. And so that would be the negative log of the Ka, gives us what's called the pKa. And this is what we look at when we want to determine whether or not an acid is on the stronger side or the weaker side. And so the larger this number is, the Ka, this number, the smaller its pKa, because these are inverse of each other. And the smaller the pKa, the stronger the tendency to lose a proton, or the stronger the acid. And so if we're talking about, say, a very strong acid like HCl, it would have an incredibly tiny, very, very small pKa. For other acids that are weaker acids, there can be a range of pKa's. Okay, so let's look at this when it's placed on a chart. Here we're talking about acetic acid, which is a weak acid. It does not completely ionize in water when you put it into water. And so what that means is, depending on the pH of the surrounding solution, this molecule will either hang on to its proton or lose its proton, depending on the pH. And so let's start all the way down at the bottom at the lowest pH and work our way through this titration curve to see what happens to this molecule as pH changes. So if you add acetic acid to water, the pH of the overall solution will be low. So that's going to be like just below 3. What this means is the amount of acetic acid that is holding on to its proton is greater than the amount of acetic acid that is lost its proton. So remember that in this environment at a low pH, there's lots of hydrogen ions already around and very few OH ions to grab hold of them. And so this molecule will be encouraged to hang on to its hydrogen proton. There's nothing encouraging it to bind to something else. There's not enough OH in the solution to encourage this to come off. So prior to titration, all of the acid is in its protonated form. It's holding on to its hydrogen proton. And so it's in the HA form. As we start to add OH equivalents, so OH equivalent just means, the amount of OH it would take to fully titrate this acid would be 1, because there's only one H on that molecule that can come off. And so if there's only one ionizing group, then it would take 1 full OH equivalent to completely remove that hydrogen proton. And so I've kind of skipped over the middle here, but I just want you to make sure you understand what this axis is telling you. So as we start to add OH groups, the pH of the solution is going to increase. We're adding OH to that. And so as you add OH groups to that, there will be less and less hydrogen, free hydrogen in the solution as it combines with the OH that we're adding. So what's going to happen then is added base will cause the protons to dissociate from its acid until it's fully titrated over here at the endpoint. And so as we move to a higher pH value, we'll start to see this molecule disappear and this molecule increase. Eventually, we're going to get to a point where they are equal to each other. And so that would be the midpoint, where the amount of acid exactly equals the amount of conjugate base. And when that's the case, that is where we have reached the pKa of this acid. And so that means the pKa is defined as the pH at which the amount of acid and conjugate base in solution is equal. That is an important definition because it will keep coming up. So I'll say it one more time. The pKa is defined as the pH at which the amount of acid and conjugate base in solution is equal. So in the context of acetic acid, the pKa of acetic acid is the pH at which the amount of acetic acid is equal to the amount of its conjugate base. And so the pKa here is somewhere below 5 because that's the pH at which we have equal amounts of these two things in solution. Okay. As we continue to add more and more OH, now on this side of the curve, we're going to get a situation where there's far more conjugate base than there is acid. There's lots of OH in this solution. Lots of the hydrogen ions are being consumed up by those OH ions. And so the H will be more encouraged to come off of its molecule and combine with those OH groups. So it's essentially this molecule and OH are competing for hydrogen ions over here. Once we have added one full OH equivalent, what that means is we've added enough base to fully neutralize the acidic component of this weak acid. And so since there's only one ionizing group, one place where there can be a lost H group, that means we are adding one OH equivalent to fully titrate that. Okay, now I want to draw your attention to this purple center place in the middle here. Note how the line for, oh wait, hold on. There we go. So note how the line here is steeper than the line here, the slope of the line here, and then becomes steep again on the other side. So when the line is steep, that means adding very small amounts of OH will have big effects on the pH of the solution. But here, when the line is less steep, that means adding OH has very minimal effects on the pH of the overall solution. And so when we're in here, this is what's called the buffering range or the buffering zone. And so what buffering means is the pH of the overall solution will not move too much as you add OH. And so we can add more and more OH here, but the line, the pH of this solution doesn't change all that much until we get outside the buffering capacity of this weak acid. And so once we're outside the buffering capacity, then adding a very small amount of OH will have very dramatic effects on the pH of the overall solution. So let's talk about this buffering zone. Oh, and I should mention that buffering occurs 1 pH unit above and below the pKa of the acid. We will definitely come back to this when we talk about amino acids. So again, buffers are really important in biological systems. Aqueous systems that can resist changes in pH when small amounts of acids or bases are added are really robust and stable systems. And so think of it this way. In our bodies, we are constantly doing different stages of cellular respiration. And cellular respiration will create hydrogen ions as a byproduct. Those hydrogen ions have to end up someplace. And what they're often ending up is the bloodstream. The bloodstream is a physiological pH, and in messing with the pH of the bloodstream is going to really affect enzyme activity for cells that are sitting in that environment. Enzymes, as I discussed at the beginning of this, have optimum pHs, and outside that pH, enzyme activity will slow or stop entirely or completely stop functioning. And so if we have the byproduct of natural cellular process like cellular respiration is creating acidic byproducts, then we have to have a way to consume those acidic byproducts enough so that the pH of the blood doesn't dramatically change every time a cell finishes cell respiration. And so what we need in our bloodstream is some kind of buffer. And so the presence of a buffer in our bloodstream will stabilize any changes in pH when things like hydrogen ions are added to the system or OH ions are added to the system. And so dumping a bunch of hydrogen ions in the system should have a really dramatic change on pH, but because of the presence of a buffer grabbing hold of those hydrogen ions, we don't see a dramatic change in pH, and the bloodstream remains at a stable, constant pH. And so buffers are very important for many different biological systems. And I'm going to come back to the bloodstream as an example at the end of this module. Okay, so this means that the concentration of your acid in solution, the concentration of your base, the pKa, or the ability of that acid to lose its proton, all are related to the pH of the system. And we can relate all of these terms using the Henderson-Hasselbalch equation. And you will have to know how to use this equation, the problems that will help you practice this concept. And so the Henderson-Hasselbalch equation relates pH of a solution to the pKa of an acid in that solution, so the ability of that acid in solution to lose its proton. Also, it's related to the concentrations of that acid and the concentration of its conjugate base. And so this equation is really versatile. It can allow you to predict the pH of a solution when given the concentrations of acid and base in that solution. It can help you calculate this value at a given pH. It can help you estimate the pH of a buffered solution and find the equilibrium pH. It's very versatile, can do a lot of things. It can help you predict at what pH your acid is going to lose its proton and so on. And so we're going to practice this in the recitation section and relate it to different biological systems. And so let's talk about one biological system here, which is the bicarbonate buffering system that is in your bloodstream. Again, I mentioned as a byproduct of cellular respiration, you create a lot of hydrogen ions. You also create a lot of carbon dioxide as a byproduct of cellular respiration. And so in our bloodstream, we have molecules of bicarbonate in our bloodstream. And bicarbonate is the buffer of our blood. Bicarbonate, when combined with hydrogen ions, forms carbonic acid. So this is formed from dissolved carbon dioxide and water. So dissolved carbon dioxide and water together can make carbonic acid. And then when carbonic acid loses its hydrogen ion, it can form bicarbonate. But this molecule, carbonic acid and bicarbonate, exists at an equilibrium state within our bloodstream. And so there's plenty of both of these in our bloodstream. And it's balanced. So that's what keeps our bloodstream at a constant pH, is the balance between these two molecules. Okay, so the pH of the bicarbonate buffering system depends on the concentration of these two molecules, which sort of oscillate back and forth depending on the needs of the body. That oscillation depends on the concentration of dissolved carbon dioxide, which depends on the concentration of gas in the lung. And so if this increases, if you have a lot of carbon dioxide gas in the lungs, then that's going to mean you're going to have more carbon dioxide dissolved in the blood. When that combines with water, you'll have more carbonic acid dissolved in the blood. And then that pushes the equation towards bicarbonate and dissociation of that ion. If you have a lot of hydrogen ions in the blood, that's going to encourage the combination of those hydrogen ions with bicarbonate, and it's going to push the reaction in the opposite direction. So that's going to make more carbonic acid. That's going to push for more production of water and carbon dioxide, and it's going to push for production of gaseous carbon dioxide, which you then breathe out. And so we are constantly oscillating between these two things by the process of expelling carbon dioxide in the lungs and the production of hydrogen ions as a result of cellular respiration. And so let's say you just started exercising, you just started going for a run. And so that exercise is going to produce a lot of lactic acid as a byproduct. So this is one way in which we can perturb the pH of the blood. The production of lactic acid is going to put a lot of excess hydrogen ions in the blood. And so lots of hydrogen ions is going to automatically push the reaction in this direction. And so this is why when you exercise, your respiration rate, or the rate at which you expel carbon dioxide, goes up, because you are pushing the reaction towards this pole. So lots and lots of hydrogen ions means you're going to end up producing gaseous carbon dioxide, which you then expel. As you expel that again pushes the reaction back in this direction. You're going to make more of these. As you continue to exercise, it pushes it in this direction, and so on. And so we have a constant pushing and balance of these two things in your bloodstream, which keeps the pH of the blood at a constant rate. And so any major changes in hydrogen ion concentration would have major changes in pH, which would disrupt cellular functioning if there was no buffer present. But because there's a buffer present, major changes in hydrogen ion concentration have no real effect on the pH of the blood.